Welcome to Chapter 7. This is going to be the cellular basis of inheritance, and we are going to build upon what we talked about in Chapter 6 and compare some of the stages of mitosis with the stages of meiosis. So let's get started. These cute little guys are just showing you that multicellular animals can begin with one single tiny fertilized cell and grow into these beautiful flamingos, these big giant hippopotamuses, hippopotami, I don't know. Anyway, or the cholla plants. I think that's a cholla plant in letter B. So everything starts out with at least one cell. And we move on to these great big characters, and we will talk about how that happens in just a minute. I know this may seem a little bit overkill, but I wanted to just quickly review from chapter 6 so that it's fresh in your mind, so that as you're learning about meiosis, you can see the difference between it and what we learned in chapter 6, which was mitosis. Okay, so let me just give you a quick rundown. Homologous chromosomes, those are two chromosomes that look alike. They have the same length and shape. They have the same genes in the same location on each of them. And remember, homo means the same, so homologous is the same chromosomes here. Now, remember, we talked about sister chromatids. Picture the letter 11, and those are two chromosomes. If you pinch them in the middle, they sort of splay outward at the top and at the bottom, right? Making an X shape. So picture that X shape attached, the two of them attached together are called a centromere. And that little movement of pinching them with the centromere creates this X-shaped figure. Well, once we get our chromosomes together, we start the cell cycle. And again, remember, this is all mitosis for right now. So the cell cycle, which we call interphase, has the three components, G1, S, and G2. And think back to that little round pie chart. G1 is where most of the growth is happening in the cell. S phase is where we get the DNA replication. It's one of the most important phases of our lives. And so we have to make sure that the S phase is always properly done, that there's no mutations or anything like that. G2, the cell doesn't spend much time in G2. It usually goes from S phase, slips right past G2, does a little bit of growing along the way, and then ends the interphase, but will start immediately into the mitosis. Also remember that there are checkpoints that indicate to each phase when to do their job, basically. So if you are at G1 and S, you need a checkpoint here to see if the cell should continue to grow or if it should stop and then move into the S phase and start DNA replication. So there's a checkpoint between G1 and S, and then there's a checkpoint between G2 and mitosis. Okay, there's not one with S and G2 at all. Remember, checkpoint is just to say, yep, we're not ready for you to go yet, or yes, we let you pass and go ahead and do your thing. If the checkpoints fail, then some major mutations are happening. So if a mutation occurs or a checkpoint fails, G1, the growth stage, could just go on and on and on forever, just like we talked about with Henrietta Lacks. Remember, her cervical cancer cells just divided ad infinitum. Really, they just go on and on and on and on because they're still growing today, and it's been about 60 years later. So that's an important checkpoint. And I like to use her story as an illustration of that, so hopefully you'll remember. Um, Moving on to mitosis, remember there's 4 phases that we talk about. And just keep in mind that the book has a pro-metaphase that it's putting in together with the prophase. And I don't like that. So we're going to go with 4 phases because that's the normal way we discuss them. So we're going to have 4 phases. They are, well, we'll talk about what they are in the next slide for a sec. I'll come back to that. One of the most important things to remember about mitosis is that it's for asexual reproduction and that it maintains the chromosomal number. So the number of chromosomes it starts with, the parent cell has, will be the same amount that the daughter cells have. And every 2 daughter cells, they each have their same. Then finally, we have cytokinesis, which is the act of actually breaking apart those 2 daughter cells. Because up until this point, at the very end of mitosis, those sister cells, no, let me say that again. The daughter cells that are produced in the very last phase of mitosis do not come apart until cytokinesis happens. Cytokinesis has to put a contractile ring around the center. Remember we talked about a balloon that you put a ring around and it tightens again and again and again until they burst apart. That's how you get, that's all that cytokinesis is. Basically, you're cutting apart your cytoplasm. All right, don't forget to know that what happens in each phase of mitosis, what the chromosomes are doing, what the spindles are doing, where they're located in each phase. That will all be important. And then we lastly talked about cancer. Cancer can arise when checkpoints don't do their job. So if checkpoints fail, we can end up with a cancerous situation. Sometimes we just end up with a benign tumor or lump on our skin. Unfortunately, as you age, you find more of these lumps here and there, lumps and bumps. My poor mom, she's discovering quite a bit of them. And sometimes they're just like little fatty tumors that go under your skin, and they're just annoyances. They don't hurt or anything. They're just, not that greatest thing to look at. If you ever had a Labrador, Labradors are, it's common for them to get fatty tumors, and they're benign, but they're just kind of unsightly. They're these big tumors that hang off of them. So they can easily be taken out if need be, if they're getting rubbed on too much or something, but you can always leave them in because they're benign. Okay, so that is our review. Let's move into chapter 7 and start comparing mitosis to meiosis. So before we get too far into this discussion, let's outline some ideas here. So first we start with the definition of a chromosome. It's a structure consisting of DNA and associated proteins which carry out part or all of a cell's genetic information. So again, we're going to be, this is sort of the precursor of learning about the actual components of DNA. So right now we're at chromosomes, which again are DNA that are wound tightly into these long stick figures, if you will, and then when they're combined at the center with the centromere, they make that nice X shape that we're used to seeing. The human body has 23 chromosome pairs, 46 individual chromosomes, and they're numbered 1 through 22 on a karyotype. If you remember that, there's a picture of it in this presentation, but it's the slide that looked like all the different colored lines, if you will, and then they line them all up together by length and whatever genes there are in a specific order. They'll all line up and pair up together, so they're all buddies and they all look alike. So the first chromosomes, they will be the same. The second chromosomes, they will both be the same. They'll go on and on like that through 1 through 22. The 23rd chromosome are your sex chromosomes. The rest of them are your somatic chromosomes. They are just everything but the ones that make babies, basically. And so the 23rd pair will either be an XX, which is female, or an XY, which is male. And those are your chromosomes for your sexual reproduction, gametes. Okay. Homologous, remember homo means the same, so homologous chromosomes are members of a pair of chromosomes with the same length, shape, and genes. Okay, so that's why they look like. That's how they pair up when they're on a karyotype situation. One member of a homologous pair was inherited from the female parent and one from the male parent. So we get genetic information from each of our parents. And every species has their own chromosomal numbers. So humans have 23 pairs, and a redwood tree might have, I don't know, 14 pairs. There are some that only have 3 or 4 pairs. So every species has a set amount, and that is unique to their species. This is just a nice little picture that I like to show so that you can have a better idea of what's happening on each individual chromosome. So here are 2. They're pink and blue, one for information coming from the mother and one from the father. And the bands that you see all across the length of these chromosomes, those bands are genes. And as it notes, they occur in pairs on the homologous chromosomes. So these chromosomes are going to be identical. However, if you notice, look at the green ones at the bottom. The pink chromosome has a brighter green color than on the blue chromosome. The blue chromosome sort of has a pale green color. And so yes, they look different, but they are the same gene. It codes for the same thing in your body. So let's say this is your hair gene, and everybody doesn't have the same color hair. And so what happens is you can get this variation from your genes, so that will indicate what color hair you're going to have. So let's say this green, both of these green chromosomes, or excuse me, the green genes are for hair. They denote hair color. And so the brighter green one on the female chromosome could indicate blonde hair, and the paler green one on the blue chromosome could indicate red hair. All right, so there's a slight difference there, but they're both indicating color of hair. When you have a slight variance like this, we call those genes alleles. So if you look at the top 2, the blue ones, the blue genes, those are homologous to each other. They look exactly the same, but they're not alleles because there's not a slight variation here. There's not a variation happening at all. They're exactly the same. I would say the same for the white ones, but then I would say that the yellow ones, the one on the left on the pink chromosome is a little more yellow, and the one on the blue chromosome is a little more orange. So I would say the the yellow and the green chromosomes, I'm sorry, I keep saying chromosomes, the yellow and the green genes on the chromosomes can be said to be alleles for whatever genetic trait those genes are for. So hopefully that makes sense. So allele is a variation of a gene. You'll hear that again and again. I'm sure you'll hear about it in lab as well. I like this picture because it just sort of outlines everything nice and neatly. So here you have your single chromosome. It goes through interphase. During S phase of interphase, we get this duplication, and at the same time, they end up pinching together in the center by a centromere, and now you've got this nice chromosome that we're used to seeing. Okay, when you see an X-shaped chromosome, that's actually two chromosomes together, right? They're your mother chromosome and your father chromosome. They've been duplicated, so they're right there together. When they're grouped together like this, pinched in the middle, you can say now that they're chromatids of each other, or sister chromatids. Okay, so the top chromatid is a sister chromatid to the one on the bottom, and vice versa. It's just another way to notate what is happening. So if I say, oh, look at the sister chromatid, you can have this picture in your head of, oh, that's an X shape. There's two chromosomes that are identical, and they're attached in the middle with a centromere. I know what those are. So that's why there are a lot of different terms for seemingly the same organ or process or whatever it is, structure. During mitosis, we're making an exact copy of our parent, and as such, during this process, during all the phases that we've talked about, there's a complete set of chromosomes that are being copied and placed into two new daughter cells. Okay. In G2, the chromosomes consist of the two replicated DNA molecules attached at the centromere, so it's making that X shape with the sister chromatids. And then when they're pulled apart, each will become an individual chromosome again in a new nucleus. So that's how we can take one parent cell, duplicate it, pull that X apart, and put half of it in one daughter cell and half in the other, and that's how they both become identical to their father, or parent, I guess you could say. Okay, and then of course we have the cytoplasmic division that will cut them in two and neatly package everything together. This photo really shows a nice depiction of what we just talked about in the last slide. So here's your father cell, or I keep saying father cell, parent cell. And you can see that there's two chromosomes in there, one from your father and one from your mother. They are duplicated in the S phase. So in this picture, you don't really see the duplication happening, but it's in between both of the first and second cell. Okay, so we've duplicated it. Now we've got two of the father chromosomes, two of the mother chromosomes. When the chromatids, so if you're looking at this, the blue X is a chromatid and the pink X is a chromatid. All right, so when the chromatids get pulled apart, one will go into each of the daughter cells. So that blue X, half of that's going to go into the top cell that you see here, and half will go into the bottom cell that you see. And the same thing happens with the pink chromosomes that were duplicated. Half will go into the top cell and half will go into the bottom cell. And so now when you look at it, you can see both those daughter cells look exactly like the parent cell. And so that's how we maintain that chromosomal number. If we didn't duplicate it in the S phase, then you would have something completely different. You would have half of half, half of these. So you would have half a blue and half a pink, and half a blue and half a pink in the daughter cells, and it would be all messed up. So this is how we maintain the chromosome number. It's critical that they duplicate during S phase. So another quick review. This is how mitosis works. Remember, there's 4 phases: prophase, metaphase, anaphase, and telophase. Each one of those, it's important to understand what's happening inside each one of those phases. So prophase, you've got the nuclear envelope breaking up, spindles are forming. Spindles will grab onto the chromosomes that have now been packaged together in nice little X shapes that we're used to. Prophase is also the first phase where you can actually see the chromosomes, but they will be all pushed together in the center of the cell during metaphase. In anaphase, they start to be pulled apart towards the polar ends of the cell. In telophase, they finally reach the polar ends of the cells, their opposite ends, and they're ready for the cytokinesis to begin, because they're separated far enough apart where when cytokinesis happens, there's going to be enough room without cutting up those chromosomes by accident. A mnemonic that's common for this is Paul met Anna at the theater. I use PMAT. That just works for me. Use whatever works for you, but mnemonics are a great way to learn all of these terms, as well as flashcards. And once again, I'm going to tell you, get together in your groups, have study groups and study sessions regularly. Read through the stuff at least half hour every night, or even 10 minutes every night. Just to keep it all in your brain. Okay, so this is the beginning of what mitosis is doing. The next couple slides are going to finish it up. So here is the rest of mitosis. I couldn't fit it all on this slide, but the only thing that's missing is prophase, which is on the previous slide. So prophase starts mitosis, and then we go right into the metaphase, anaphase, telophase. And then again, of course, we always do the cytokinesis, which will be illustrated on the next slide. Here's another depiction that we've been talking about for the cytoplasmic division. This is the end of the whole mitotic phase. So you have the contractile ring that is starting to wrap around the center of the 2 chromosomes, and then it pulls in tighter and tighter, making a cleavage furrow, which is like the little dip inward, and then it just keeps going until pop, there's 2 new cells. You know, you got these 2 little eggs with eyes staring at you. Okay, so that review was a little bit longer than I was expecting. Now we're getting into the new material for Chapter 7. So before we get started, though, we need to talk about sex and alleles. Okay, so asexual reproduction is what mitosis is. Okay, sexual reproduction is meiosis. So that's a big distinction right there. Mitosis in asexual reproduction ends up with 2 diploid daughter cells. Meiosis in sexual reproduction ends up with 4 haploid daughter cells. And I'll explain what all that means in just a little bit. Asexual reproduction has offspring from one parent, and the offspring are genetic clones. They're identical to the parent. Asexual reproducers are less adaptable to environmental changes. If there's an entire species living in an area where the environment changes, becomes harsh or inhabitable to the species, they will die off very quickly unless they find a new niche that's acceptable to them. But because they can't adapt as well, they will die off. So it's important, in a way, to have your species scattered about, because if you're all huddled in one place and you have an environmental change, then they're going to just be completely wiped out and now you're extinct. So spread out. Sexual reproduction, you start with a parent cell. That parent cell has genes from two parents, your mother and your father. Okay, so you've got genes that are going to be passed down to the children, and those genes are coming both from the mother and the father. Sexual reproducers are hardier. They can live, or I should say, they can adapt to environmental changes fairly readily, and they have a good chance of surviving in such situations. So the species isn't really in danger in that respect. Sometimes we get these mutations that happen, and they will spread quickly throughout the species, because if your mother or father had a mutation, then they had sexual reproduction happen. Obviously, if you're talking about humans, they're going to have sex, but let's just talk about sexual reproduction in this cellular level here. You're going to pass on your genetic material to your offspring, and each offspring is going to have that same genetic makeup that you have, but now the parent has a mutation. That mutation is going to be passed on from child to child to child. Every child's going to have this mutation. Okay, they're always passed down. Okay, if you have a mutation, it doesn't mean that you're going to have an ill response to it, a disease from it. It just means that you have a mutation and there's a potential for something to happen. And oftentimes that mutation is not something that's beneficial. There are some mutations that are beneficial, and we need mutations in our genetic lines because that's how we get genetic diversity. It's one way we get genetic diversity. Okay? And then finally, again, I'll just recap. Sexual reproduction, there are 4 daughter cells and they are haploid. And I'll explain what that means in just a bit. Diploid means 2. So there's 2 daughter cells that have 2 chromosomes that are exactly like the parent. Okay? So let's move on and to get into more detail about meiosis. Okay, let's really get into the meat of this meiosis stuff. And again, I'll be going back and forth, sort of comparing it with mitosis as we go along. Somatic cells, those are the ones that are any cell other than a gamete. A gamete are your reproductive cells. So your XX, which is female, or XY, which is male. Meiosis happens in the gametes. So there's your difference, another difference there. Mitosis happens in your somatic cells, so every cell other than your gametes, and then meiosis happens in the gametes. These are your reproductive cells. You're going to be producing children with these cells. Okay? Meiosis halves the parental chromosome number by sorting chromosomes into 2 new nuclei twice, and we call it meiosis 1 and meiosis 2. And duplicated chromosomes of a diploid nucleus, which is a 2N, that's how we write it, are distributed into 4 haploid nuclei. So go back to mitosis for a second. Mitosis began with 1 diploid parent cell and ended up with 2 exact copies, so they would be 2 diploid daughter cells. Conversely, meiosis starts with a diploid, or excuse me, a haploid cell, and when it is finished, you'll end up with 4 haploid cells. Okay, haploid cells are one set of chromosomes, denoted as a little n, and then diploid has 2 sets of chromosomes, 2n. And just remember, diploid has 2 because one comes from the mother, one comes from the father. Okie doke. So the ending of mitosis and meiosis are different, and they're basically the important part of both processes. So mitosis ends in and 2 daughter cells that are diploid, and the meiosis ends in 4 haploid cells. So they're haploid, they have 1 set of chromosomes. And that's not 1 chromosome, it's 1 set of chromosome. And diploid has the 2 sets. Here's a quick outline of what meiosis is. So we still have interphase, where remember the DNA is replicated in the S phase right before meiosis. So instead of going from interphase to mitosis, now we're going from interphase to meiosis 1. Meiosis 1 is identical to mitosis. So if you've already learned mitosis, you know what meiosis 1 is. Okay, so we still have prophase, metaphase, anaphase, and telophase. And then once we get down to that telophase, we move over and repeat the whole process again and call it meiosis 2. And again with prophase, metaphase, anaphase, telophase, all number 2s. One important note here, notice how interphase is just before meiosis 1. We do not do a second meiosis, or excuse me, a second interphase before meiosis 2. So from interphase we go into meiosis 1. From meiosis 1 we go into meiosis 2. From meiosis 2 we go into cytokinesis. Okay, which isn't shown here, but that's the outline. Now we're going to get into the heart of it on the next slide. Okay, so here we're starting with meiosis 1. So interphase has just ended. We had that nuclear, we had the chromosomal duplication, DNA duplication. We've moved on to meiosis 1. This is going to have still going to have your nuclear envelope breaking up, the chromosomes being packaged into that X form that we like. With sister chromatids, we're still going to have the spindles lining up the chromosomes, and then the spindles pulling those chromosomes apart and putting the parts into each cell that is created, basically. And so at the end of meiosis 1, we have 2 cells that have formed. Because remember, this is a lot like mitosis, okay? So we've got 2 new cells. Then we move into meiosis 2. We divide it again, which are separating those sister chromatids, and basically go through all those stages once again. But now you've got 2 cells that are going through meiosis 2, and the result ends up in 2, each one of those ends up in having 2 more cells. So at the very end, you're going to have 4 haploid nuclei, and it's going to go through cytokinesis and it will all break open. Okay, and so make sure that you look at the slides that have the pictures, because I think that's going to make it have more sense than me just kind of explaining it to you. If only you could see my hands. I'm Italian and I need to speak with my hands and it makes things make sense. Okay, moving on. We briefly mentioned this a little while ago. One of the cool things about meiosis is that the genes get all mixed up. the alleles, remember those alleles on the chromosomes? Those genes or alleles get all shuffled and mixed around in various different ways. And so we get an allele, which is a variation of a chromosome, or excuse me, a variation of a gene on the chromosome, or we get some of the genes are removed or they're duplicated. It just depends, but we get this shuffling and this mix-up of genetic combinations, and that's one way how we get genetic diversity. And so we want this to happen. We want to have genetic diversity. It makes us hardier as a species, as an individual. We don't want to all look the same. Otherwise, we'd be like little robots. We don't want the same skin tone. We don't want the same hair color. We don't want the same mannerisms. So one way this happens is through this process called crossing over. This happens in prophase 1, and then there's this thing called random assortment that happens in metaphase 1. So crossing over is where you have an X-shaped chromosome, or we call them sister chromatids, that sits as close as it can get, sometimes just right on top of a second sister chromatid. Now think about that for a minute. There's two X's lying on top of each other. The components of each X are sister chromatids to each other, right? So they've got like a left hand and a right hand side, right? And the big thing here is that the two sister chromatids, those two X-shaped chromosomes, are not sister chromatids to each other. And so that allows them to cross over, swapping their genes. So the genes from one will go to the leg of another one, and the gene from that one will go back to the first one. And again, you need to see my hands. No, I'm kidding. There's another picture coming up that you can see this. So that's crossing over. Random assortment is where basically what it's just saying, it's going to happen in a random way. We don't know which way the genes are going to fall. Sort of like crapshoot, you throw dice and you're not even quite sure how, what's going to come up. Russian roulette is kind of like this. You get this random assortment. Okay, and so again, that contributes to our diversity. Here's the depiction of crossing over that I was talking about. You can see, so you've got a blue chromosome, sister chromatids, and the red chromosome, sister chromatids, right? They get close enough to each other where they cross over. Then at the bottom there, you can see where they've swapped their genes completely. Okay, so go back to the first picture. Notice how on the blue, each side of the blue has a capital A, capital B, and a capital C on both sides. The red one has lowercase a, b, and c on both sides. So when we cross over, there's a potential here to have a shuffling of those genes. So then we go to the bottom picture. Here we have now the recombinant crossing over. We've got the lowercase c on the blue one now, and the uppercase C on the red one now. So we've switched genes, we've swapped them, and it's done randomly. It could have been an A and a B, or an A and a C. It could have been any combination of those 6 letters on each one. So that's the part that's the crapshoot that you're not sure of. So we sort of flip a coin and hope for the best. This is another more animated picture to show you what we've been talking about. So you can see we've got sister chromatids in the red X and in the blue X, but note that the red and blue are not sister chromatids of each other. Okay, you have to be the same to be sister chromatids. In prophase, they start the crossing over, and they make what they call a tetrad. You do not need to know the word tetrad. From here, it goes through several different processes to actually make that crossover happen and stay where we've crossed things over. And that's what the endonuclease, the DNA ligase, and the recombinase come in and what it means there. You don't have to know those three words either. So just know that you go from the prophase 1 is the crossover, metaphase 1 is the recombinant, assorted recombinant genes. And so you can see here where they've switched little legs of each end and flip-flopped them. So this is crossing over in meiosis and results in the genetic diversity. The next couple of slides are going to show you in detail what happens in each phase of meiosis. Just like we did in mitosis, this is almost identical. So remember, we have interphase, G1, S, and G2. The checkpoints have passed us through. Nothing's happening that shouldn't be, and we move right into meiosis 1, starting with prophase 1. Remember, crossing over happens in prophase 1. The nuclear envelope starts to break up in prophase 1. The chromosomes start to package themselves into the little X shape that we're used to, the sister chromatids. And the chromosomes are now visible, whereas before in interphase you couldn't see them. Okay, spindles also start to form in the cell. Okay. Metaphase 1, this is where the recombination happens. You've got a pair of homologous chromosomes, you can see in that little picture there, and then you've got a second one. The spindles will line up the chromosomes all together in the center, just like in mitosis. This happens in metaphase 1. In anaphase 1, the chromosomes are starting to be pulled apart, and then in telophase they're completely pulled apart at the opposite ends of the cell, and they are starting to take this shape of an actual cell. So they're starting to get this circular outline happening, but notice that they are still attached together. All right. Also note how each cell has a small X chromosome and a large X chromosome. Okay, and they're different. So the small X on the top is pink and the big X on top is blue, except for its little foot there. And then the ones on the bottom are opposite. Okay, so you've got a little bit of recombination happening here that you can see. Let's look at the next one. So once meiosis 1 is done, we move into meiosis 2. So basically the process is repeating again, but it's taking those two cells from the end of meiosis 1 and we're starting with two of them this time. So in meiosis 1 you start with one cell and end up with two. In meiosis 2 you start with those two cells that you created in meiosis 1, and you end up, as you go through the process, with 4 cells at the very end. Okay, you get 4 haploid cells. Alright, so you go from 2 cells to 4 cells, and this is how it's done. So if you go into prophase 2, here are your 2 cells with the 4 chromosomes all packaged together. The spindles are starting to form. Nuclear envelope is starting to break up again. Remember, there's no DNA replication happening here. Okay, we're just taking those same 4 chromosomes that we created and we're working with those. We're going to line them up in metaphase 2 like we normally do. In anaphase 2 we're going to pull them apart like we normally do, and then you're going to end up with 2 cells on the top and 2 cells on the bottom. You can see that, right? Okay, so look how everything's been distributed. There's a tall blue chromosome in each cell on the top. there's a tall pink chromosome in each cell on the bottom. And then it does the same thing with the little cells, the little chromosomes. So we've got little chromosomes on the top that go into each cell, one in each cell. Notice where the recombinant one is, so that's going to be a little bit different. And then you have the bottom one doing the exact same thing. So you've got all things happening here that are all over the place basically. So you end up with 4 haploid cells, and they're haploid because they have 2 chromosomes in each one. Okay, we started with 4 chromosomes in each. We ended up with 2 chromosomes in each. All right, so from here it will go through cytokinesis again, like we've talked about. There's a lot going on in this slide, but it's really great, so take some time to look through it all carefully. This is like a one-stop shopping overview of meiosis. So starting with number 1 at the top, we go from diploid to haploid. That only happens in meiosis. Okay, mitosis is diploid to diploid. Meiosis is diploid to haploid. It shows you here metaphase 1, telophase 1, metaphase 2, all diploid. When you end up finally with telophase 2, you end up with haploid cells. Okay? At the bottom, it's showing you how the crossing over happens, again only in meiosis. Have diploid cells in prophase, where the crossing over happens. Your random assortment is in prophase, or excuse me, in metaphase. That's the random assortment there. That only happens in meiosis. Then you go on to anaphase 1, then which is diploid, prophase 2, which is diploid, and then finally anaphase, which is diploid, and then you end up with telophase 2, 4 haploid cells, or you can say that we have 4 gametes. We have formed 4 gametes in the meiosis process, and that's our goal. And so you can see those 4 at the very end, and it has 1 chromosome from the mother and 1 from the father. Okay, hopefully that makes it make enough sense for you. For this slide, we're going to talk about gene segregation. So think about that for a minute. Go back to what you know about your sister chromatids, what they look like. You should be picturing the letter X. And then in meiosis, we had crossing over and random assortment. Here we're going to talk about more how we get the genes into the gametes, basically. All right, so we start with those homologous chromosomes. They separate during meiosis, and so the pairs of the genes that they have will also separate with them. And then at the end, when you get to the gamete part, the gamete will carry 1 copy of the 2 members of each gene pair. It's a difficult way to think about it, but basically you're getting 1 gene from your mother and 1 gene from your father. And so this is showing how that happens. So if you look at the first 2, or I should say the first column with the 2 cells, they're in metaphase 2 because they're all lined up. Be able to recognize that. In the center column, if you look at the top half of that first cell, you can see a pink chromosome folded over and a blue one folded over. Then at the bottom of that cell, you have another pink one folded over and a blue one folded over that has a crossed over pink tail or foot, however you want to think of that. Now look at the 2 cells right next to it. There's a short pink one and a tall blue one in the first cell. The second one down has a small pink one and a tall blue one with the recombinated foot, right? So that though both of those came from that top cell in the center, right? That one cell made two new gametes. And then the exact same thing happened with the bottom cell in that center column. It put the chromosomes into the remaining two gametes. So you've got a short blue one in each of the gametes and a tall pink one in each of the gametes, with the tall pink one with the blue crossed over foot. Okay, so this is a nice little depiction of how we get those four gametes and how they end up with one from each parent, a chromosome from each parent. Okay, and each one of those chromosomes have all of the genetic material on them from your parents. So all the genes that your dad had, all the genes that the mother had, those are on those cells in those gametes. This figure is in your book. It's a really great figure if you can read it. It's a little bit small, and get out your magnifying glass or put on your reading glasses, which is what I've had to do, and squint your eyes really tightly, and maybe it'll all make sense. We'll work with the top half first. In the gray column on the left, it says chromosome arrangement 1. So this is one possibility of a combination of the genes. So basically this is saying, here are the four chromosomes that we start with, and you're each going to end up with two chromosomes, and this is these are the possible combinations that we can do to mix them up. Okay, so let's start at the top. Metaphase 1, everybody's lined up in the center. So you can see tall X's and small x's, reds and blues. The blue ones go to the left, the red ones go to the right. They've done their thing, metaphase 2, and then they separate out into your gametes. You have two blue in each of those gametes. One's large, one's small. And then in the other one, it's one large, one small. Then the same thing happens with the red cell. The red split into two new gametes. We've got a big red and a small red in both of those. So that's one possible combination of the gametes, of the chromosomes, of the genes. All those things are kind of the same. Now, for the box below that one, it says chromosome arrangement 2. So this is a different possibility. Okay, these are scenarios that can possibly happen to help you illustrate and understand what's going on here. So take that top circle again, and instead of having the same colors split out, we're having the cell on the left with a big blue and a little red, and then the cell on the right with a big red and little blue. Now we have a whole new mix going on. Let's see what happens when we get to the gametes. The gametes possible layout or division would be like this. You have two tall blue ones and then two small red ones on the left. Then on the right you have the opposite. You have two large red ones and two small blue ones. Okay, so two different possibilities or scenarios of how we can do this random independent assortment thing happening, and how we end up with the variation at the end when we have the gametes. In this figure, we are comparing what happens in pro-metaphase 1 to anaphase 1 and pro-metaphase 2 to anaphase 2. So basically we're comparing meiosis 1 with meiosis 2. And for our purposes, remember we're only talking about 4 stages of meiosis. Okay, the prophase, metaphase, anaphase, and telophase. So says prometaphase, and you're probably thinking, well, are we going to consider it prophase, or are we going to consider it metaphase? In this instance, we are considering what's happening here. The very first portion, we're going to say, instead of prometaphase 1, we're going to say metaphase 1. And the same goes for the second one. We're going to say, instead of prometaphase 2, we're going to say metaphase 2. Okay, and then anaphase is going to stay the same no matter what. So, in metaphase 1 of meiosis 1, you have 2 pairs of homologous chromosomes. So remember, the blue ones, both blue ones together, that's a pair of homologous chromosomes. The red 2 together are a pair of homologous chromosomes. All 4 together, the 2 blue ones and the 2 red ones, are not homologous to each other. Okay, so only the 2 colored ones that are the same are homologous chromosomes. Okay. In metaphase 1, both pairs are lined up in the center of the cell. Remember, they're pushed there by those spindles. Okay. From metaphase 1, the spindles will move into anaphase 1, and those spindles are going to start separating the 2 homologous pairs. Okay. And so they're still attached in the center at the end of anaphase 1 by the centromere. Okay. In meiosis 2, what's happening is that we have 1 pair of homologous chromosomes. You can see that crossing over has happened because there's some blue at the tips of the chromatid on the left, at the top and the bottom, and we still have these microtubules attached. And what's going to happen in anaphase 2, those spindles are going to pull those sister chromatids apart by those spindles. Okay, and they're going to be the spindles come out of the kinetochore, or those centromeres, basically. The center of the cell is called the chiasma, and you don't really need to know that term, it's just that's what it's called. Okay, so that's a difference in what's happening in metaphase 1 and metaphase 2. Okay, in metaphase 1, we have 2 pairs of homologous chromosomes that are being separated in anaphase 1. And I failed to mention, at this point, when they get to anaphase 1 and they're being separated, crossing over has happened already. Okay, so between the metaphase 1 and the anaphase 1, the crossing over happens, and then they're pulled apart. And that's, as you can see, the blue pair stay together and the red pair stay together. In the meiosis 2, in metaphase 2, you have 1 pair of homologous chromosomes that has already had crossing over. They're pulled apart, and now we have 2 separate chromosomes. Okay, one has variation of genes in it, and the other one has the original genes. And so those are the differences between meiosis 1 and meiosis 2 for these 2 stages. This figure, 7.6 in your book, is a really great one, and I wish it was bigger so that you could see it. What I recommend doing is taking the actual PowerPoint that I have posted, not the video part, but the PowerPoint itself. Go to this slide and copy this figure and put it into a Word document, and then you can make it as big as you need to fully see it. You might even be able to do that directly on the web version of the textbook. So play around with it, see what you can do. But for my eyes, this is a little small for me to read, but I think we can get through the gist of it. Basically what this is showing us is it's comparing meiosis with mitosis, and that's what's happening in the first 2 columns, or rows, I guess you could say. And then the little chart at the bottom that has more squares in it, it's showing you, again, comparing meiosis with mitosis, and at the top of that little figure at the bottom there, it says process, and so it'll tell you the process and what the differences are in that process between meiosis and mitosis. Okay, so we're going to kind of go through this as best we can. Let's start with meiosis, which is the very top box, the yellow or pale tan, however you see it, the very top one. That says meiosis on the left, and it starts with interphase. So it's showing you interphase here. Interphase, if you can see it, it's a little bit squiggly in the center there. That's your DNA. Technically, you would not see the DNA at this point. You would see it in prophase. We'll just kind of run with that. If you notice, again, we're talking about prometaphase there in the center. So it went interphase, prophase 1, prometaphase 1, and then metaphase 1. The prometaphase here in number 1, consider it metaphase. So there's going to be a lot happening with this metaphase. So think of prophase as the nuclear envelope is starting to dissolve. The spindles are forming and they are starting to attach to the chromosomes. All right, and the chromosomes now in prophase are X-shaped. They've tightened and coiled together, compacted tightly, so that they have this X shape now. They're gathered at that centromere to make the X shape, and then the prometaphase 1 we're going to consider metaphase 1. Those spindles have pushed those chromosomes, those X-shaped chromosomes, into the center of the cell, and you can see that in metaphase 1 there. So in this case, they're showing you the in-between little bit here where you've got prophase and there are all the X chromosomes, and they're going to be pushed together, which is what prometaphase 1 is showing you, and then metaphase are all lined up completely nice and neat. For our purposes, again, prometaphase, just consider it metaphase. So metaphase in this count is the spindles are pushing the X chromosomes to the center of the cell, and that's what we see above the metaphase 1. All right, moving on. Anaphase 1, those spindles are starting to pull apart those X chromosomes. Now, be careful here. These X chromosomes are staying in their X shape. They are staying in their homologous chromosomes to each other, right? You can kind of see here where at the top of the anaphase cell, it's got a red X and a blue X, and then below that it has a blue X and a red X. Okay, so you can see they've been pulled apart. The 4 groupings have been pulled apart, and you still have 4 homologous chromosomes. Okay, then in telophase, the separation is starting to happen a little bit more. So you can kind of see where in telophase the outer membrane of the cell is starting to indent a little bit right between or right in the middle of that cell horizontally. So you're going to have 2 homologous chromosomes on the top and 2 on the bottom. Okay, it's not really detailing this, but you can kind of see right after telophase we have cytokinesis, which you can consider that gray bar, that gray line there. That's cytokinesis. And then you can see after cytokinesis in meiosis 2, we have the 2 new daughter cells that happened at the end of telophase 1 in meiosis 1. Okay, so at the end of telophase, we are getting new nuclear envelopes around both sets of chromosomes. All right, and then it's indenting a little, and in cytokinesis, it indents all the way. It separates them completely. We have the 2 new daughter cells that begin meiosis 2. All right, so now those 2 daughter cells begin prophase 2 in meiosis 2. And then everything just repeats itself again. In metaphase 2, the spindles are going to push those two homologous chromosomes now into the center of the cell. Okay, do you see that? They're all lined up nice and neatly. There's a blue homologous chromosome on the top and there's a red homologous chromosome on the bottom. That's at the end of my metaphase 2. Okay, then we move into the telophase 2 where the spindles have all broken apart. So you can see broken apart each homologous chromosome, I should say. And you can see at telophase 2 we have a cell that's beginning to indent in the middle. It looks like it's getting a little waist, you know, like you're cinching a waist. It's got one homologous chromosome split in half on the top and the bottom. And so we have, if you look at the cell between metaphase 2 and telophase 2, it makes a little more sense. You can see how the homologous chromosomes have been pulled apart. So now on the top of that cell, we have two chromatids that have been separated. We've got blue chromatids that are separated and red chromatids that are separated. Remember, chromatids are the parts of the X that were pushed together. Okay, let's go back just quickly for a minute. Think back to what a chromosome looks like that we talked about originally. It looks like a straight line. If you put them side by side, it looks like the letter 11, the number 11 that is. So if you have the number 11 in chromosomes, then those individually are called chromosomes, right? When they attach at the middle with the centromere, then that number 11 now becomes an X shape, and each side of the 11 that was gathered, the original 11th, those become sister chromatids, right? So we have half of the X on the left and half of the X on the right, and each one of those halves, the left and the right, are now called sister chromatids or chromatids to each other, however you want to think about it. So what we're having here in metaphase 2, we had our two chromosomes, our sister homologous chromosomes, the blue X at the top and the red X at the bottom. In the middle there you can see how the X's are pulled apart. So we've gone back to having the number 11 essentially. It's just here it's showing it bent in half. So each number 1, if you will, is bent in half. Okay, so you've got the blue chromatid on the left and the blue chromatid on the right, and then below that you have the red chromatid on the left and the red chromatid on the right. And then finally at telophase, where it's starting to show the waist cinching of the cell with the contractile ring, all 4 of those are going to be separated. All right, so, and if you'll notice, that's all come from one daughter cell. So I'm going to back up just a little bit. We went from interphase with one cell into the prophase with one cell. Once we went all the way through meiosis 1 and cytokinesis and the beginning of mitosis 2, we have 2 daughter cells at the end of meiosis 1 that start meiosis 2. That top daughter cell is going to produce 2 new daughter cells. The bottom daughter cell is going to go through all of meiosis 2, just like we did on the top, and end up with 2 new daughter cells. Okay, and if you can see them, now we've got 1 blue and 1 red chromosome in each daughter cell. So now we have a chromosome from our father and a chromosome from our mother in each daughter cell. Okay, so that's how we get our parental DNA from both parents into each cell. All right, it's a long process. We have to start with one cell, go all the way through meiosis 1 and get 2 daughter cells, and then go all the way through mitosis 2 and get, or excuse me, meiosis 2 and get 4 daughter cells. Okay, and we're going to back up one more time. Okay, if you'll notice, those 4 daughter cells now are haploid. So in the beginning, we start with a diploid cell. Okay, it goes all the way through and we end up with diploid daughter cells. Those daughter cells go through meiosis 2 and we end up with 4 haploid cells. All right. The diploid indicates a homologous chromosome. So you got to know what a homologous chromosome is. A homologous chromosome are those 2 individual chromosomes that look like the number 11 that were joined in the center. Now they're X-shaped. That X shape is a homologous chromosome. If you have 2 X's, 2 homologous chromosomes, in this case we've got a blue and a red in one cell. If you have that, then you have a diploid cell. When you only end up with one chromatid from one X and a chromatid from the other X, in this case one red, one blue, one mother, one father, in each cell, that makes it haploid. So we've gone from like 4 chromosomes basically, and they're joined in various ways, down to 2 chromosomes. So 4 chromosomes or 2 haploid chromosomes, or excuse me, 2 homologous chromosomes go from that all the way down to the 4. So we've got diploid and ending up with 4 haploid cells. Okay, and I know I'm going on and on and on about this, but it's very important that you understand this process. Okay, now skip down to the next box. This is mitosis. And what we're doing here, see how we're skipping? We don't have anything under what would normally be above meiosis 1. We're skipping all of that. We're going to start with our cell in interphase. We've gone through our DNA replication just like we did above in meiosis. We're skipping all the way down, and now we've got one daughter cell, or one diploid cell that we're starting with. So it's got the 2 chromosomes, excuse me, 2 homologous chromosomes. The same things happen in metaphase. Those spindles push those chromosomes to the center. In anaphase, they start pulling them apart. In telophase, they start separating them again, and then cytokinesis ends up popping them out, and you're left with 2 chromosomes, or excuse me, 2 daughter cells at the end. So prophase in mitosis is the parent cell or the father cell, however you want to think of it. At the end of cytokinesis in mitosis, you have 2 daughter cells. Those 2 daughter cells are diploid. In mitosis, this is a big difference from meiosis. In mitosis, we start with 1 diploid cell and we end up with 2 diploid daughter cells. Okay, so mull that over a little bit. Think about what you've just learned in meiosis and how that compares with mitosis and why we've pushed it all the way down to the second meiosis area. Okay, why we don't have anything written under meiosis 1. So just think about that for a little bit. It's important to know whether you're starting with a diploid or a haploid, and it's important to know whether you end up with a diploid or haploid. Very, very important, very distinct differences, okay? Remember, mitosis is asexual reproduction. Meiosis is sexual reproduction. The 4 haploid cells that you end up with in meiosis, those are gametes. Those are your sex cells. They have sex chromosomes in them, okay? So that's where sexual reproduction comes about. In mitosis, the daughter cells are exact clones of the parent cell. Okay. Now, this bottom column here, or table, again has the processes that go across the top. Then we talk about meiosis in relation to each one of those processes, and then we talk about mitosis in each one of those processes. So the processes are DNA synthesis, synapses of homologous chromosomes, crossing over, homologous chromosomes line up at the I can't read that, but in the center of the cell basically, and sister chromatids are pulled apart is the next column, and then the outcome is the last column here. The outcome says number, and honestly I can't read that, number of something components of daughter cells. So how many chromosomes basically are you going to end up with? Okay, what's the final count in the daughter cell? What do the chromosomes look like, and what are inside those daughter cells, and what are they called? So going back to the beginning, meiosis, DNA synthesis occurs in the S phase of interphase. Oh, look, it also occurs in the S phase of interphase in mitosis. The synapsis of homologous chromosomes happens in prophase 1 in meiosis and does not occur at all in mitosis. Okay, crossing over happens during prophase 1 in meiosis, but does not occur in mitosis at all. Homologous chromosomes line up at the metaphyseal junction or plate, whatever they want to call that. That happens during metaphase 1, and again does not occur in mitosis at all. Sister chromatids line up at the metaphyseal plate. That happens during metaphase 2, and in mitosis it does occur. It happens during metaphase. Remember, mitosis just has one metaphase. It doesn't have a metaphase 1 and a metaphase 2. It just has a metaphase. And our final result at the end of meiosis is we get 4 haploid gametes, or 4 haploid daughter cells, however you want to think about it, at the end of stage 2, at the end of meiosis 2, that is, okay, at the end of cytokinesis, at the end of meiosis 2. They don't show cytokinesis here in this little table. In mitosis, you end up with 2 diploid cells at the end of mitosis, which is really at the end of cytokinesis that happens after mitosis. So don't forget about cytokinesis. It shows cytokinesis up above in the figure with each cell, but it doesn't talk about that in this little table, so don't forget that part. Okay, now hopefully all of that made sense. Mull it over, read through it again and again, and get out your magnifying glass and see if you can read that. And again, copy and paste it into a Word document to make it bigger. I think it's the only way you're going to see it well, at least if you have eyes like mine, which hopefully you don't. You're all young and can see pretty well, I hope. Okay, let's move on to the next slide. This is another karyogram or karyotype that we've talked about before. It's figure 7.7 in your book. Again, this is just a typical tool. It's an easy, quick glance. It allows a quick glance by a geneticist to quickly go, oh, look, everything here looks uniform. I can run down the line of 1 through 22 and see that they're all uniform. It doesn't look like any of the genes or the alleles are out of place, and so we have this nice healthy person with no genetic defects. And then of course it also has the gametes listed, which are technically your 23rd chromosomes. In this case they are both X chromosomes, and so remember, X's mean female. All right, and if this was a male, it would have 1 X and 1 Y. All right, so if you were asked how many chromosomes does a human have, how many pairs of chromosomes, you're going to say 23. Okay, you're not going to say anything other than 23 in a normal, healthy person. Even though these karyograms only show 1 through 22, plus an X or an XX or an XY, however it does that. It's not going to be 22. The answer is 23. Okay, we have 22 somatic cells with these chromosomes, somatic chromosomes, and then we have one pair of gametes. All right, or one sex cell, however you want to think of that, sex chromosomes. This slide is going to talk about some defects in the genetic makeup that can happen. So we've talked about this karyogram in the previous slide, and again, that's a tool that the geneticist will use to quickly glance at each one of your chromosomes to see if there are any obvious problems that are being easily looked at. Some of the things that can happen have to do with your sex chromosomes only. Okay, and so this one is an example of your X inactivation. So if an X chromosome goes awry, something happens to it, in this case it inactivates, and it only inactivates one of the X's. So let's say this is a female. It's going to inactivate one X chromosome. Well, this person still has another X, and that X will still make you female, and all your chromosomes should still be normal in appearance. Whereas genetically, there's like a little bit of a defect happening here. So this says, in this case of the cat, the embryonic inactivation of one of the two different X chromosomes that encode for coat color, and encode just means there's a gene for hair color or coat color. Encoding means that the gene is working. Okay, so we've coded it, or we've encoded this different coat color, and they will give rise to this tortoiseshell phenotype in cats, obviously. Phenotype, we'll talk more about that in the coming chapters, but phenotype means it's what your outward appearance looks like. So it's everything that somebody, when they look at you, this is what they see. So when we look at this cat, we see the really pale, kind of freaky eyes. He's not very happy. That doesn't have anything to do with phenotype, but he's not very happy because his ears are all wonky. But his coat color, it has this nice tortoiseshell, beautiful appearance, and that happens when there's an X chromosome that's been inactivated. Okay, hopefully that makes sense. So basically something had turned it off. An error in the environment or whatever it was has turned this X chromosome off. Okay, it says one chromosome is inactivated in each cell into a structure called a bar body. All right, and so it's just another term to say, hey, this is where that chromosome is stored now. It doesn't work, so we're going to package it away neatly into this little body, I guess you can call it, or box, or however you want to think about that, compartment. And then the genes on that chromosome are not expressed in the individual or any of the individual's offspring. Remember that mutations are passed down to offspring. Okay, and that mutation is in every single cell because of the way meiosis happens, right? Go back and think about that with that chart. The chromosome that inactivates is random, so there's no way to know, hey, this one's gonna inactivate, or 90% says this one's gonna inactivate but not that one. It's totally random. Okay, there's no way to know. It's a random deal. So this is called embryonic inactivation. And again, this happens early on when there aren't a lot of chromosomes and things in the mix yet. There's not a lot of development going on. There's not a lot of cells that have been produced. Okay, in the next few slides, we're going to be talking about human genetic disorders. Okay, again, this would be something a geneticist is going to determine, and oftentimes these are the errors in our genetic code that we are concerned with about passing on down to our children, and so we will go and list a genetic counselor who is a geneticist, and they will use a karyotype. They're going to use some other tools we'll talk about in other chapters, such as a pedigree or a Punnett square. They have tools at their disposal, and oftentimes it's just a blood test that they're going to look at all of your DNA. So what they will do is they will get the results. They'll discuss them with you. They'll say if there is a genetic defect or disorder, defective genes, there's a mutation, there's a deletion, there's inactivation, whatever is happening, they're going to give you all the information, and then they're going to give you all of your options. And basically your options are to move forward with the pregnancy and get as informed as you possibly can along the way. So when the baby's born, you'll know how to deal with any issues that are pertinent to that genetic problem. You may give up for adoption. You know, who knows what you might do. You might keep the baby or give it up for adoption. Your other option, of course, is to terminate the pregnancy. And that is a valid option in all of these cases. It's not to say that it's what everyone would choose. Obviously, everyone wouldn't. You know, and it's not one of these things to be in an uproar about. This is science. We're talking about science. Yes, there is great emotion dealing with these situations. Of course, if this is you, you're not going to go, well, I'm a scientist and I'm going to take away all my emotion. I'm going to look at this as objectively as a professor would and just go from there. Of course, you're not going to do that. However, the most informed that you can get is going to be the best way to help you make a decision. And that decision is yours and yours alone, and it's very personal, and there's no right and no wrong. It's whatever is right for you. And of course, this is my opinion, and how I feel the world should work, basically. So yes, this is my opinion. And I would never say, oh, you should not have done that choice. You know, that if that was right for you, that was right for you. All right, so moving on, we're talking about genetic disorders. Move down towards the bottom of the screen. Let's look at these definitions first before we talk about the top part. Here's some definitions for you that they kind of talk about in the book a little bit. We'll talk definitely more in the next few chapters, but I want to give you definitions here so that you understand what these sentences above are talking about. Okay, so remember we talked about an allele. Picture that single chromosome with all of those bands on it. Those bands are your genes. And if you have your second chromosome with all those same bands and they all match up and they're all aligned, then you don't have any issues. But if you have an allele, which is an alternate form of a gene, then you have a variation happening in whatever that gene is for. So we've talked about before hair color. If that gene has an allele on one chromosome, then for hair color, then the hair color is going to be slightly different. Or you have the potential to have something different. Okay, so like if that gene is for hair color, one allele might say it's brown hair and one might say it's blonde hair. Okay, so the gene is hair, but the allele indicates that there are alternate possibilities. We've got the possibility of getting blonde hair, brown hair, black hair, red hair, white hair, you know, any of those colors. Okay, so the gene is for hair color. The allele indicates that there are several options that that gene could produce in your outward appearance. Okay, dominant. We've all heard of this. These are traits that are inherited unchanged in a hybridization. Okay, so hybridization means you're taking genes from one person or parent or plant, as we'll discuss in the next chapter, and you're taking genes from another individual. Okay, so you've got two individuals that are contributing genes to this one offspring. Okay, and if that happens, you're going to have dominant traits and you're going to have recessive traits. The dominant traits are the ones that you're going to see in a phenotype. And remember, phenotype, and I'll put that on there, phenotype is your outward appearance. Okay, so dominant traits are those that are inherited, unchanged in a hybridization. They're the ones that you are going to see in a person or in that individual, depending on what species we're talking about. And recessive traits are the latent traits in an offspring of that hybridization. So latent just means that they're sort of under the radar. They're still there, but they're kind of, you don't see them, you don't hear them, you don't, you know nothing about them other than they're there, but they're not going to be doing anything. Okay, so it's like they're in timeout. They're in the corner, you're not doing anything with them. So dominant traits you see, recessive traits you do not. Homozygous, remember homo means the same, are genes that have two identical alleles. Okay, remember, alleles are alternate forms of a gene. Homo means they're the same. These are same alleles. So if we have a gene for hair color and we have two identical alleles, then that hair color is going to be the same color no matter what. In essence, it's kind of the dominant trait. Heterozygous, hetero means different or not the same. And again, zygous just means an egg or a cell or something. Heterozygous is a gene that has two different alleles. So if it's hair color, then it would be blonde and brown, let's say. Okay, so homozygous, two identical alleles. Heterozygous, two different alleles. We have dominant traits that are seen or expressed, is often a term that you hear, and recessive genes are the latent ones. Okay, now let's go back up to the top. Human genetic disorders, remember that's what we're talking about here. Some dominant or recessive alleles on autosomes. Remember, autosomes are the ones that are everything but the sex chromosomes. Okay, so some dominant or recessive alleles on autosomes or the X chromosome are associated with genetic abnormalities or disorders. So basically what this sentence is saying is any one of your chromosomes can have a defect, okay, can acquire a defect or a disorder or an abnormality. It can be on a dominant one or a recessive one. Those are your only two options, dominant or recessive. And it can be on an autosome or an X chromosome. Those are your only choices, your gametes, which are your X chromosome or Y chromosome, or the rest of them, which are your autosomes. So basically it's talking about in a complicated way, every single one of your chromosomes have the potential to have a genetic abnormality or disorder. It doesn't mean you're going to, but you have the potential for something going wrong. Okay, and in later chapters we'll talk about what cause mutations a little bit more in depth. So next sentence, an autosomal dominant allele is expressed in homozygotes and heterozygotes. So whether you have two identical alleles or two different alleles, one of them is going to have a dominant factor, okay? And in this case, they're talking about autosomal dominant. That means it's your somatic cells. It's everything other than your gametes. Okay, so your chromosomes 1 through 22. If any of those have a dominant allele on a homozygote or heterozygote chromosome, or alleles, excuse me, the dominant one's going to be the one that you see. The dominant one is expressed. Okay, so that's what that's meaning. The autosomal recessive allele is expressed only in homozygotes. And here's why. If you have a homozygote, they have two identical alleles. They both can't be dominant, and they, well, I take that back. They both can be dominant, and they both could be recessive. It's not likely. Usually it's one or the other. So and you have a homozygote. They have the same alleles, the same gene for those alleles. One's going to be dominant and one's going to be recessive. Okay. In homozygotes, the only way to express, I should say this, the only way to express a recessive allele is if it appears in a homozygote. So let's back up a little bit. Homozygotes are genes that have two identical alleles. If those alleles are identical and they happen to be recessive on your autosomes, then they're going to be expressed because there's no dominant trait right there. So recessive is only going to be the only thing there because it's homozygote, which means that both of them are recessive. In the sentence above that, the autosomal dominant allele can be expressed either in a homozygote, where that means both alleles are dominant, or in a heterozygote where you have a dominant and a recessive. Then, of course, the dominant is the only one that you're going to see. The recessive just sort of hides underneath that dominant one. You'll never see it. Okay. This does get complicated, it gets wordy, so I want to kind of break it down for you here. Let's see, so autosomal recessive, autosomal dominant. Again, autosomal, we're talking about everything but the sex chromosomes here. Okay, so we'll learn more about this when we get to Punnett squares in the next couple of chapters, but let me detail something for you that might help make this make sense visually. If you have a homozygote, and we're talking about gametes, okay, I know before we weren't, but let's just talk about gametes for a minute. Remember, gametes are your sex cells. If you are talking about a homozygote, then your genes are either going to be XX or XY. No, I take that back. You can only have, I meant to say XX or YY. Well, there is no such thing as a YY. A YY does not mean male. Male is XY, okay, and XX is female. All right, so if we're talking about homozygous gametes, that means they're going to be female because they're going to be the same, XX. Heterozygote is an X and a Y. Okay, they're different. They have two different zygotes, two different chromosomes. They've got an X and a Y. Homozygous have XX. Okay, so kind of picture it like that. Homozygous has XX, they're going to be female. Heterozygotes are going to be XY, and they're going to be male. Just when we're talking about the gametes. These next few slides are going to talk about different types of disorders. It's going to give you some names of them and some symptoms that you might see. And as nursing students or allied health students, you may run into people with these issues. So, autosomal dominant disorders. Autosomal are anything but your sex chromosomes, right? And dominant means it's going to have a dominant allele. The dominant one's the one that's going to have the trait that's going to be expressed. Okay, so here's in the left column disorder or abnormality. In the right column, you're going to have symptoms of those disorders. So the first one, achondroplasia, the main symptom is dwarfism, and we're probably all familiar with this now because we see it on TV all the time. So if you watch the TLC channel, you see they've got like 3 or 4 different shows of people that are dwarfs. They like to call them little people in their shows, from what I understand. The technical term that science uses is dwarfism. Okay. Camptodactyly, this is where you have, and I can show you on my own fingers, which is why I wear my rings that I have. Camptodactyly is rigid or bent fingers. And so like if you were to stretch your fingers out straight, this would mean that the very tip that last joint by your fingernail is going to be bent downward. Okay. It also means that the rest of your finger is very straight. Okay, it's very tight. Sometimes it can't be moved. Sometimes that middle knuckle there gets a little bit hyperextended. Okay, and so I can show you exactly what I mean on my own fingers when you see me in lab. That's why I have the rings on, to keep me from hyperextending and from keeping that bent finger happening. Now, I don't necessarily have this disorder. I have something completely different, but symptom is the same in this particular case. All right, so familial hypercholesterolemia. Let's break this word down. And if I haven't told you before, if you ever get the opportunity to take a medical terminology class, jump on it. That class will be invaluable to you. If you can break down parts of a word and understand what those parts mean when you put them together, then you can understand what doctors are talking about. If they're throwing out fancy terms or something, you could write it down and go, oh, okay, I know what this part means, I know what this part means, and I know what this part means. So put them all together. Oh, okay. This guy, in this case, is having trouble with his cholesterol. His cholesterol is elevated. So familial hypercholesterolemia. Hyper means excessive or high. Cholestro means cholesterol. Emia means a condition of the blood. And so this particular thing, familial hypercholesterolemia, comes from your family members, so it's passed down. High cholesterol levels, and it means in the blood, because like when you go and take a blood test, they can test your cholesterol. So that's where the emia part comes in. That's why it says in the blood, or a condition of the blood. Huntington's disease is a degenerative disease of the nervous system. Marfan syndrome is abnormal connective tissue. This particular disease is actually a sister disease to my own. I have what's called Ehlers-Danlos syndrome, and my connective tissue is super lax, and so I have really hyperflexibility, and I get dislocations and things like that. Marfan syndrome is very similar. The body types of people with Marfan's are very similar to my own. When you think of Marfan, the key characteristics are, yes, abnormal connective tissue, but when you look at somebody, you can kind of say, hey, I wonder if he has this, because they're going to be very tall, and they're going to have long limbs, long legs, long torso, and they're going to be very thin. Their face is going to have a long stature. They think Abraham Lincoln had Marfan syndrome. And I read an article around the Olympic times, a few Olympics ago, I guess. I think it was the one in China, where Michael Phelps was swimming and he won all those medals. At the time when they were interviewing, they had mentioned that he has been tested for Marfan syndrome. Now, when you think of Michael Phelps, you see that same body type. Very tall, lanky, long body, limbs, legs, face, facial features and things like that. Turns out there is a genetic test for this, so he was able to get the test. It said it was negative. The tests are not that great, and so every now and then he has this test repeated. And you might say, well, what's the big deal? Well, he might not be showing symptoms of problems on his body if he has Marfan's, but later on in life, he may find effects from this. Another characteristic symptom of this, like my own, is nonstop, all over body pain. And the pain is quite intense, and so that might be something that he needs to know, that he can prepare himself for, that he can be looking out for, new kinds of pain medications, and what would work for him, what wouldn't. You'd have a long talk with your doctor and all that kind of stuff. Okay, so that's why these genetic things are important, is because you might not be showing symptoms now, but later on you might, or later on you might not, but your offspring might. So it's important to have these genetic tests done. Polydactyly just means there's an abundance of digits. So we've got extra fingers and toes here. If you've ever had an orange tabby cat, you probably noticed that they had 6 fingers or 6 paws, not paws, 6 toes. That's a very characteristic symptom or thing that you would see on an orange cat. Progeria, and there's a picture that we'll show you about this one. Progeria is a rapid aging. You get this drastic premature aging, it says. Neurofibromatosis is tumors on the nerves and in your skin. So if you looked at your arm, especially around the wrist where you already have those bones that pop up, they're a little bit, kind of round. You might have on the nerves in your arms or in that area of your wrist, you might have little bumps that pop up, and they can sometimes be very painful. Other times they might just be bumps. Sometimes you can have neurofibromatosis without tumors, but you might see like a cafe au lait spot on the person's skin, so it looks like somebody spilled coffee on you. That's where the cafe au lait comes in. They're usually quite large. A lot of times they're found on the backs of the calf muscle, calf of your leg. And so I know when my son was little, we had him tested for this. He had a cafe au lait, and then what they did is they measured the cafe au lait, and they looked for other types of markings on his body. They looked for moles or other small little brown spots all over him. And if he had 13 or more of a combination of these, then they would consider him having neurofibromatosis, but they would also do other tests. There's an eye exam that you could have that will show it. And I forget, there's some other tests for that. So a lot of times there's multiple tests for each thing. And again, just if you have a genetic disorder doesn't mean you're going to have problems with it. Okay, just means that you have the potential to have problems. It also means that you're going to pass it on, but it doesn't mean that your offspring are going to have problems. It just have the potential to have those problems. Here are some more disorders. These are autosomal recessive. So the other ones we saw were dominant traits. These are recessive disorders. So these are going to happen on homologous chromosomes. Because remember, that's the only way that we can have recessive traits being expressed. If there's a dominant trait in there, it's going to be overshadowing what that recessive is. So autosomal recessive disorders are those on homologous chromosomes. I know a little less about these than I did the other slide. Albinism, pigmentation, or actually these are not very lined up very well. It's albinism is a disorder, and the symptom is absence of pigmentation. Okay, so those lines got a little off kilter there. Let's see if I can figure out this next word. It's methemoglobinemia. Okay, so I don't know what methemo is. I'm guessing that it's blue because I can see what the symptom says. Globin, I'm guessing, is the shape of the cell, or something in there is round, is what I'm guessing. And then emia is a condition of the blood. So there's a condition in the bloodstream that's causing blue skin discoloration. That was my guess. I don't remember what all of those parts mean, so I knew the last one. All right, cystic fibrosis. Very horrible, tragic disease. I don't like this disease. Not that any of them are good, but this one's particularly awful. Abnormal mucus secretions. And what happens is the secretions build up in the lungs, and as your lungs fill up with fluid, it becomes harder and harder to breathe, and over time you eventually essentially drown in your own secretions. It's just tragic. There are machines that you can put on. They put on like this vest that's sort of like the one that, like a life safety vest that you put on in a boat. It's similar to that. It's plugged into a machine and it's turned on and it jiggles you. And it just jiggles your body up and down, up and down, up and down for however long it's set for. And it's to break up those little mucus secretions so that they can be excreted by the system, by your body. Okay, so people don't always live all that long with this disease, but as these tools and these drugs and everything improve, as we find better technology, we have people living longer with diseases. So we're able to handle them better. So you can Google cystic fibrosis. If you were in my ground class for lecture, I would show you a video of this lovely young lady. She was 16 and she demonstrated how her vest worked, and she was just adorable. And so you're looking through the comments, of people saying, oh, good luck, bless you, or whatever. At the end, her family had written, thanks to everybody who had commented because they were mostly positive, and she had died later that year. So it was really, really sad. So I try to show videos of things that I think you might run across in your field. And so cystic fibrosis is unfortunately becoming more common. So you might see that. A lot of these are not as common. I made the comment one time in my ground class that, albinism, it's very rare. We're not going to see this, and, probably never in our lifetimes are we going to meet somebody that has this albino trait. And I had a young lady from Africa in my class, and I think she was from Kenya. I can't remember exactly where she was from, but somewhere in Africa. And she said, oh, in my village, albinism is very common. We have, a large population of albino people. And I thought, oh, wow, that's really, really interesting. So it also illustrated to me that I get into my own little box, and my own American little box, that I can't think past my American ways. And I want you guys to think more globally, because we are becoming a global nation instead of, every country, because we can get places in an instant, through planes and technology. We can talk to people all around the world at any time of day. So we're becoming this global nation. So it's important to understand what's going on out in the world outside of our own comfort zone. And so that one kind of opened my eyes. And I thought that was an interesting comment. And I was really appreciative that she pointed that out to me. Okay, moving on. Ellis Van Creveld syndrome is dwarfism. So we've got another type of dwarfism here, and they usually have heart defects. So this dwarfism is a recessive disorder. The other one, achondroplasia, was a dominant disorder, and they are both dwarfisms. They're going to look differently, and then different organ systems, I guess, are more affected than in one than the other. All right, so then Fanconi anemia. Fanconi, I'm assuming, is the person who discovered this. Bone marrow failure. So your bone marrow is not producing red blood cells. And so your hemoglobin is low. Your hemoglobin is what carries the oxygen, so you're not going to be breathing well. You're going to be anemic because you're not going to have as many red blood cells, and you're not going to be producing them. The next one, galactosemia. Galacto is a liver term. It doesn't mean liver, but it pertains to the liver. And then again, we have emia, which is blood disorder. So this one also affects the brain and gives you eye damage, unfortunately, and the liver, of course. I'm assuming that you could probably get a liver transplant, and perhaps in the one prior you can get a bone marrow transplant, and perhaps that would be sufficient for you, but I don't know that for sure. Phenylketonuria is also called PKU. It's pretty common. I mean, not everybody has it, of course, but from my limited understanding of it, it has to do with your kidneys even though it's giving you mental impairment, it's a kidney disorder, and your kidneys are sort of riddled with these holes. And I think you can also have the opposite. You can have cysts as well. I don't remember entirely, but it's a urine or a kidney problem, but it gives you mental impairment. So it's affecting your kidneys are basically kind of working at less capacity, and so the nerves that go from your brain to your kidneys are not working well. But from what I understand, also it happens more frequently in women than in men. Sickle cell anemia. This is another one that I learned something new about. We often, as Americans, understand this one to be an affliction of African American people. And again, not all African Americans have this. If they have it, it's a genetic disorder, just like this is showing you. It's an autosomal recessive disorder. And it says adverse pleiotropic effects. And so that means that it's going to affect many body systems, and there's too many to list. What I have learned in the past, again from a student, is that people from Africa, people that live in Africa, I should say, the population of people that live in Africa, the majority of them have sickle cell anemia. They have sickle cells. These sickle cells, while they would be detrimental to African Americans, let's say, it can cause some effects with diabetes, for one. The Africans that have sickle cell anemia, they may still have all these pleiotropic effects, but one positive thing that happens is it keeps them from acquiring malaria. And as you know, in Africa, there are a plethora of mosquitoes. I mean, more than you can probably ever count in a lifetime. And they all carry disease, and mostly they carry malaria. And so over time, we've had this population of Africans that get sickle cell anemia, and it protects them from acquiring the malaria disease. Basically, you're not going to get sick from it. You may still acquire it, but you're not going to get sick from it. And I thought that was really, really interesting. And so there is a purpose for this sickle cell over time, and we have evolved, that population of Africans had it evolved to have this sickle cell in them, and it helps them thwart malaria, which is prevalent in their environment. Whereas we have it, we don't have as many mosquitoes. We still have mosquitoes with disease, but not on the scale that Africa has. And so instead of showing that protection, that positive thing that happens, because we don't have malaria really in America, so we're not going to see that, how, hey, they're not having effects from it. We're going to see, oh, they have sickle cell anemia, they're going to have diabetes, and we're going to see all these problems, because that's what we have in America. So again, think globally and think about these things. Always read global studies from other countries on the same disorders that you might be looking at. You find a lot of really interesting things. There are some disorders that are chromosome number specific, and we're going to outline a bunch of these here. So let me give you a term first of all. Aneuploid or aneuploidy indicates that an individual has an abnormal number of chromosomes. So again, when I say in a healthy person, how many chromosomes do we have? You would say we have 46 chromosomes, or we have 23 pairs, right? Because 46 is 23. Don't forget to count that gametes there. So we have 1 through 23. In these cases, trisomy 13 and trisomy 18, this means that chromosome number 13, chromosome number 18 have 3 chromosomes. Instead of the pair of chromosomes, they have 3 chromosomes. They have an extra one. So trisomy 13 is also called Patau syndrome, and has this unfortunate extra fingers that come, extra appendages that are growing out of this baby's hand. They have mental retardation, motor disorders, they have trouble picking up their head, they have smaller heads, they have extra fingers, so microcephaly, ceph means brain or head, they have a smaller brain. Polydactyly, dactyly means fingers, poly means many. They often only live for a year, which is tragic. Trisomy 18 is called Edwards syndrome, and they also have microcephaly. They also have the cleft lip or cleft palate. They live a little bit longer, but, they live only 5 to 15 days. I was thinking years at first. So they live barely at all, which is even more tragic. I mean, I can't even imagine. But look at the chromosomes, you'll see that there's 3 on each one of those chromosomes. So if you have trisomy 13, you've got 3 chromosomes at number 13, and trisomy 18, you have 3 chromosomes at number 18. Here's another aneuploidy, and I think that definition in the slide before that said that it was just an additional chromosome that happens. Aneuploidy can mean either an additional chromosome or missing a chromosome. So it goes either way. It just means an abnormal number of chromosomes. In this case, we're talking about monosomy X. So mono means 1. We're talking about the X chromosome. So this is your gamete. This is the female part of your gametes. Monosomy X means Turner syndrome, and so on the X chromosome you're going to see, I believe it's an extra chromosome, or I'm sorry, no, it's 1 chromosome. It's monosomy. You only have 1 X chromosome instead of 2 if you're female. If you're a male, you don't have the X at all, I believe. It's non-fatal. You get the short stature, the swelling, the broad chest, the low hairline. You can see they don't have much of a delineation for their neck. It's very stiff and webbed, as they call it. Amenorrhea means that there's no menstrual cycle, and sterility means they can't have children. Their eggs basically are sterile. Congenital heart problems, hypothyroidism, which means a decrease in your thyroid activity, diabetes, vision, and hearing concerns as well. So again, another form of a genetic disorder. This one happens to be on your X chromosome. So here is a disorder that we're very familiar with. We hear about this all the time. What I think is cool is that over time our technology and our medications and things that our treatments have improved so tremendously, and psychologically as well, for Down syndrome, that Down syndrome children are actually growing into adulthood and they're leading incredibly productive lives. When I was a kid, my best friend had a nephew that had Down syndrome. This is trisomy 21, and so 21 means it's chromosome 21, and you have an extra chromosome. So at chromosome 21 you have 3 chromosomes, and that's what we call Down syndrome. And when I was a kid, my friend's nephew was only thought to that he was going to live maybe into his teenage years if he was lucky, because we didn't know how to handle their problems back then. And this wasn't all that long ago, 20 years ago, so semi-long, but in terms of medicine, not quite so long. And now, again, they grow into adulthood and they have very productive lives. They can have families and everything like that. And I think that's incredible. What I also think is incredible is that we are starting to see people with disorders, especially mental disorders, as people, as everyday people. We're learning to recognize that they have the same wants and needs and feelings that we all do, and that if given a chance, they can lead productive lives. And so medicine sometimes likes to limit you on things you can do, but um, if you've ever met a Down syndrome person, you'll know that they have no limitations. They don't understand limitations. They just go and do. And they're the happiest people I've ever known. Um, and I like that they're living into adulthood now. Uh, Sometimes they'll have certain physical features that we're familiar with. There's a picture on the next slide. Sometimes they have the dark circles under their eyes and they're a little bit drooped downward. Sometimes they have higher foreheads, just various things. Speech impediments and things like that. And then non-disjunction leads to trisomy 21, so it increases with the age of the mother. All right, so the older the woman is when she gets pregnant, the more likelihood she has of having this error on trisomy 21, or having trisomy 21, having the error on chromosome 21. So that's one of the reasons why they like to do genetic counseling on what they call high-risk pregnancies. Usually you're on women 40 or older. So here's that visual example of the Down syndrome. They're showing a happy young boy. You can see his eyes are little, lower lids are puffy, they're a little drooped. Their palates, I think, are a little different. They're not quite cleft, but I think they have a difference with their palate. But look at the karyotype. You can see at first glance, if you're a geneticist, you're going to look at this and go, okay, number 1 look good, number 2 look good, number 3 look good, 4 or 5, they all look pretty good. Okay, great. And if you're a geneticist, you know to look at the common ones. You're going to look at, what have we talked about so far? 13, 18, and now 21 we're talking about. So 13's good, 14's good, 15's good, 18's good. Oh wait, shoot, 21. 21 is not good. 21 has an extra chromosome, which is a total drag. And this particular offspring or child happens to be female, or excuse me, male, because it's got an X chromosome and a Y chromosome. Okay, so unfortunately this male will have Down syndrome. And in most of the things that I've read, if you have 3 chromosomes, then there's a high prevalence that the child will have the outward appearance of a Down's. You'll actually have the disease. So remember I said before, just because you have these disorders doesn't mean it's going to manifest in you. In Down syndrome, from what I've read, is that if you have that 3rd chromosome on number 21, you will have Down syndrome. Now I'm sure there are some exceptions to that, but I think there's a higher possibility of actually having that. And of course yes, they're living longer into adult lives, productive lives, but they will have health issues along the way. Okay, doesn't mean that they're going to be without any health issues. They have to deal with their heart defects and anything else that comes up. So, but they can learn to live with it just like anybody else and have those adult lives. So far we've been talking about disorders where there has been an extra chromosome on a specific chromosome number. Okay, so like the 13, the 18, and the 21, we've all had extra chromosomes. This one's the opposite. This one, we are missing 1 chromosome on chromosome number 5, and it's actually not the entire chromosome that we're missing. We're just missing a piece of it. Okay, so it's going to, when you look at it, one of them is going to be a little bit shorter, and you're going to notice, hey, these genes aren't lining up. These alleles are different. And so, oh, shoot, one's taller than the other. I better do some tests to make sure that this is what I'm seeing. And so if that is in fact the case, where you're missing a piece of your chromosome number 5, you have this disorder called Cri-du-chat. And Cri means cry of the cat, basically. Chat is cat. When these children are babies, their wailing or their crying sounds like a cat crying. which is really sad. If you've ever heard a kitten cry, that's kind of what they sound like. And this same boy was followed from age 2 to 12, and you can see the variations that happen as he ages. His forehead is kind of high. He's got something going on with his eyebrow situation, his forehead. His mouth is a little bit different. Looks like his teeth are sort of he has a larger jaw maybe on the top. It's a little hard to tell, but it looks like they're trying to point out that there's very specific physical anomalies that this child will have if you have this syndrome. And from what I understand, it's very rare. I hope. All right, we hope that all of these are rare, but unfortunately they're not. Here are your examples of the autosomal inheritance. This is the progeria and the albinism. So we talked about autosomal dominant, and progeria is an example of that. So we've got the dominant gene is overshadowing the recessive gene, and we're getting this rapid aging, unfortunately. Albinism is a recessive disorder, an autosomal recessive disorder, and so remember, for recessive traits to show, it has to be in both chromosomes. So they both have to be recessive. If there's a dominant in there, it's always going to show the dominant one. So in order for recessive traits to show, both have to be recessive. They're going to be homologous to each other. So homologous chromosomes are going to be recessive in this particular case example. Okay. Okay, now let's talk about the X chromosome. These are called X-linked disorders, and we're first going to talk about the X-linked recessive disorders. So remember, we're talking about chromosomes. If you have homologous chromosomes, you can have a recessive disorder showing, I guess you could say. And so the alleles on the X chromosome are inherited, of course, and they're expressed differently in males and females. And so remember, females are XX. We've got 2 of them. So if something goes awry with one of them, we still have one to deal with that can get us through. If you're a male, you have one X and one Y. If your X is not working, then all you have are your paternal parts. You're not going to have any of your female parts. And we all know that we have similarities between males and females. And when I say parts, I don't mean like specific body parts. It can manifest in that, but we'll talk about that later. So let's use this example. Males cannot transmit a recessive X-linked allele to their sons. Now, that's a fancy way of saying males will never pass on an X chromosome to their son, because the X chromosome comes from the female. The female has 2 X's, and they're going to give, when they pass them on, let's say they have 2 boys. So you have a father and a mother, and you have 2 sons and maybe a daughter. I don't know. The mother's going to pass on one X to one child, another X to the other child. The father's going to give them both an X and a Y, and so you're going to have one female and one male, basically. So typically the mother, well, not typically, all the time, the mother will pass on the X chromosome, okay, because it comes from her. The male will never do that. So males cannot transmit a recessive X-linked allele to their sons. Can't even do the dominant ones either, but that's neither here nor there in this slide. Females will pass X-linked alleles to male offspring. Okay, Queen Victoria, I guess, had hemophilia. I'm not quite sure why it says and hemophilia, but hemophilia is a good example. So hemophilia is what this picture is showing. You don't have specific clotting factors. Your clotting proteins are damaged or non-existent. Something's inhibiting them from clotting, and so you have a very real threat of bleeding out very quickly. A simple finger cut like this could kill you within hours because you will not stop the bleeding. You have to be given injections or IV saline, something like that. Not saline, but IV treatments of vitamin K. Vitamin K counteracts any, I think of it as loose or watery blood. So if you're bleeding out like this, you need to drink vitamin K or ingest vitamin K to help you get clotting factors to that area. Okay. And I just know that from veterinary world. If you have an animal that has drank antifreeze, antifreeze is going to cause you to bleed out in all your internal organs and orifices, and it's a miserable way to die. But if you can get vitamin K into their system quickly enough, they have a slight chance of being saved. So that's how I knew about that one. Similar to hemophilia. So these are all X-linked recessive disorders. And two examples, I guess Queen Victoria had something, maybe she had this hemophilia thing, but hemophilia is the one that we have. The main takeaway from this is that males cannot pass on X-linked problems. They can't pass on their, only females can pass on those X's, those X chromosomes. And here are some examples of the X-linked recessive disorders. So remember, these are being passed on from the female. Androgen insensitivity syndrome. This is a male with female traits. We're learning a lot about this now on our media, our social media, and our TV. There's lots of talk openly now, which I think is great. So the more we get these taboo type of things out in the open, the better we can deal with them, the better people can feel about themselves and live their lives. And so that's kind of what's happening here in the news lately. We've got male with female traits. Red-green color blindness is the inability to distinguish between red and green and other colors. Oftentimes, I think your book might have a picture of it, but oftentimes it's a matter of, for example, if you're looking at a bush with red berries, the person that's colorblind that can't see red will just see the green bush, or will see like a grayish colored, gray-green color bush. So they can't distinguish those very well. Fragile X syndrome, is a mental impairment. It has to do with the X chromosome, of course, and it's recessive. I don't know anything more about it than what it says. Hemophilia is that impaired blood clotting, like we just talked about. Muscular dystrophies is a progressive loss of function. Again, another horrible, horrible, horrible disease. Not that any of these are pleasant, again, but these are the ones that you hear in the media a lot of times. So you know a little more about them, so you know the details of them more, and you know the afflictions people have more so than maybe some of these, like the Fragile X syndrome. I've never heard of it. I don't know what it is. Obviously it has mental impairment because that's what it's telling me, but I'm sure if there was a telethon about it, I would know all the ins and outs about it. But that's kind of what this muscular dystrophy is. We hear about it all the time. Just like Down syndrome, we hear about it all the time. So we understand these things a little bit more than some other ones. X-linked anhydrotic dysplasia, which is abnormal hair, teeth, skin, and nails. So I believe anhydrotic is, I guess that would be the abnormal hair, but then it has all this, it's probably related to like the connective tissue that holds your nails and hair and skin and teeth and stuff in place. That calcium, I'm guessing, there's probably like a deficiency there of some sort. So here is the picture of color blindness example. This is again the sex-linked condition of red-green color blindness. Here we have on the left a nice green bush with red berries. If you don't have the ability to distinguish between red and green, you're going to see a muted green, sort of that gray, yellowy color on the bush, and you can't tell that there are any berries on this bush at all. And if you can, kudos to you, because I've been looking and I don't see it at all. So here is one other type of test for color blindness. They call it the Ishihara color test plate. If you see the number 71, you have normal eyes. 21 indicates you've got what they call dichromacy or trichromacy, which means you're having issues seeing, you'll either see 2 colors or 3 colors, basically. And then no number indicates achromatopsia. I don't know how you say that. Something like that. That means you only see 1 color. I actually see the number 74 in here. So I don't know what that means. But to me, it looks like there's 2 colors of the brown, actually. 2 colors of the brown outline, and I see 2 colors of the green, except for at the bottom where a 4 would be, like the corner part of a 4. That to me looks like it's more of an olive green. So I guess it just depends on how old you are and what your genes say. Okay, so this is showing you how we can get these changes in chromosomal number. We can have non-disjunction or aneuploidy. Non-disjunction is the failure of chromosomes to separate during meiosis or mitosis, and so it's showing you that here in this second picture. And then aneuploidy is a cell has too many or too few copies of a specific chromosome, just like we talked about in trisomy or monosomy. And so you can see those at the bottom there, actually in the 3rd row on the left and at the bottom row on the left, or actually on the right as well. So this is our last slide. This is 2 interesting different types of problems with chromosomes. Let's talk about A first. A says inversion, and I find this one really interesting. This is where the chromosome segment will break apart from the chromosome. It will flip upside down and then reattach itself in the exact same spot. And I just find that because these things are always moving constantly and DNA is being repaired and duplicated and all that stuff, that to find the exact same spot to me seems pretty incredible. I guess one could argue, though, that it happens so fast that it's not a big deal and it makes sense that way, but it just seems strange to me and fascinating, which just sort of solidifies the idea of how incredible the human body is. But the second one is B, reciprocal translocation, and that will occur between 2 non-homologous chromosomes. So remember, these chromosomes are not the same size and shape or length and don't have the same genes and alleles and things on them. And so you can see that before, and what happens is it's not going to cause any genetic information to be lost or duplicated, but it's still going to flip upside down. So this is also another interesting one because it's not gaining or losing any genetic information, yet what's the point to this? Is this just weird random mutation happening? Was this something that's vestigial, which just means that it's left over from a millennia ago? You know, what is the point to this one? This one's kind of interesting. So somebody do some research on that and get back to me. All right, you guys, chapter 7 was long and hopefully not too arduous for you. There's lots of information in here, so continue to go through these again and again and again. And I will see you in lab. In the meantime, email me any questions.